My name is Cher Lacoste. I'm joining you here today from a place traditionally called Stoopdale, which is loosely translated to mean stinky place. And that was from many fish carcasses in the Cypress River that is right directly adjacent to the DFO lab that I work at. So happy to be a part of this conversation today. Thanks for joining. As we see our numbers continue to climb from those on both sides of the border, always great to see that. For those of you who are new to this group, this is sort of a grassroots collaboration between stakeholders that is sort of replacing the former Salish Sea conference. So that was underway a few years before COVID, and it almost got too big for its britches and couldn't quite land on a home. And so we decided we want to continue this dialogue of issues that are important on both sides of the border within the Salish Sea. So we sort of landed on this speaker series and dialogue, which has been great and growing each time we do it. So hundreds of people joining in. So welcome to you if you're new. This was created as a forum for sharing science on an ongoing basis. So while we're still very sad about losing the World Juniors to the US, we are happy to know that we have partners on the other side of the border that have shared concerns about our waterways and our ecosystem. So thank you for joining today. So our speaker today is Greig, who is an ecosystem scientist within the DFO science branch. He's currently based at RHQ, so in Vancouver. His background's in ecology and environmental science and is a program lead of a modeling program within the regional ecosystem effects on fish and fisheries. His PhD research was conducted at the University of British Columbia and led to the Hindcast of the Salish Sea, a NEMO-based physical oceanographic model. So we're excited to have Greig join us today.
Thanks very much, Cher. That's great. And thanks to the Science Roundtable and the organizers for the opportunity to speak to you today. I believe everybody can see the first slide, and just let me know if you can't.
Looks good.
I focus, I'm focused particularly on end-to-end marine ecosystem models using a combination of oceanographic and ecology or ecosystem-based models. And in addition to leading an integrated ecosystem modeling program at DFO, I'm also a member of the Global Ocean Modeling Lab at the Institute for Oceans and Fisheries at the University of British Columbia, and that's led by Professor Ville Christensen, who's one of the key leads for the past several decades developing a framework called Ecopath with Ecosim, Ecospace. So I'll be talking to you today about this modeling work focused on the Strait of Georgia in the Salish Sea using two frameworks, the NEMO and the Ecospace frameworks. And yeah, I think almost everybody will be familiar with the area, but just in case, this is a map of the Salish Sea located in the northeastern Pacific. The major geographic areas within the Salish Sea are quite different from each other, including Puget Sound in the southeast and the Juan de Fuca Strait, which connects to the Pacific Ocean, the Southern Gulf Islands, and then the Strait of Georgia, which more or less speaking is highlighted in the darker gray box. So the Salish Sea is really, really cool. It's a semi-enclosed inland sea. It has both fjords and characteristics of an estuary. The Fraser River accounts for much of the freshwater input. And there's just generally a lot going on here. The area is temperate and is highly productive from an ecological standpoint. And a lot of the population of the American Pacific Northwest and the southeastern British Columbia is concentrated in the coastal areas surrounding the Salish Sea. So the shaded rectangles in the map correspond to two models I'll discuss today. An oceanographic model, which is the bigger rectangle, and an ecosystem model domain, which is nested within it. That's the Ecospace model domain. And then the motivation to develop the models I'll talk to you about today was to help evaluate hypotheses about the mechanisms behind declines in marine survival of juvenile salmon, and especially coho and Chinook salmon. So I'll get into that in a little more. But before I go any further, I just want to touch upon a couple of terms and concepts And so first of all, these are computer simulation models I'll be speaking about. The details vary, but the models are designed to simulate interactions and processes in the marine ecosystem. And they're especially helpful in cases where there is high complexity and a suspicion that many factors are important, not just like one or two. And the interactions represented may be predator-prey. They may also be interactions mediated by habitat or physical, chemical, or other environmental processes or factors. And the interactions include fishing and other activities. They can generate insights from scientific first principles, and they generally tend to try to build from a mechanistic and deep understanding of the system. Marine ecosystem models are also useful to explore how cumulative effects and combinations of multiple drivers of change are manifested. An end-to-end marine ecosystem model that is a kind of model that tries to include processes all the way from physics up to fisheries. So it's a comprehensive type. And a hindcast refers to a backward-looking simulation. Hindcasts recreate past conditions where there were sparse observations or maybe no observations. And the development, just generally speaking, has kind of an interesting past. And I'll just touch upon that because it helps with contextualizing it, the work today. In one thread, the main aim was to better account for interactions between species, especially fish species and their predators and prey and habitats. And according to the leaders in the field, one goal of the marine ecosystem modeling is to marry key parts of ecology with the best parts of fisheries science. And ecosystem models in this context sit next to several other types of common and useful models, and each has its strengths. But ecosystem models can be particularly helpful for answering specific types of what if questions, like what if a fishery is targeting the prey of a species targeted by a second fishery? Or what if a harvested species is also consumed by another species that has recently increased in abundance? Now, typically, these models are considered like strategic as a strategic category of model. And that means they're used to complement models that are better suited for short-term predictions or more precisely focused on tactical kinds of questions, like year-to-year fisheries management or other types of questions. But in recent years, there's been arguments made that maybe we should steer these models towards a more tactical, gradually more tactical kind of applications due to new methods to deal with uncertainty, increasing computation power, increasing volumes of data being collected, and that we should tailor it to ecosystem models that look at more tactical management questions. But um, yeah, and I think importantly too, in 2023, the Canadian Department of Fisheries and Oceans reiterated its intention to, uh, quote, speed up the implementation of an ecosystem-based approach to fisheries management in Canada and to take into account the biology and the environmental conditions affecting the stock. So the department is gradually moving towards integrating environmental variables and biological and ecological information as part of stock assessment and as just part of fisheries management. And they're developing this capacity for analysis and modeling of environmental information to support stock assessments and to just generally support fisheries management decisions. Now, I promise history lesson is almost done, but just to set the stage a little bit more, in parallel to that thread of development of marine ecosystem models, they were also developed in biological oceanography. And scientists were interested to understand the role of plankton and motivated to understand these critical feedbacks that regulate climate. For example, like carbon and nutrient cycling and the biological pump of the ocean. This led to the development of detailed and sophisticated biogeochemical models. So that's a mouthful, but yeah, biology, chemistry, geology, ecology. And there's still a lot we don't know about these lower trophic levels, the phytoplankton and zooplankton ecosystems, and how they actually work. How do they actually function? So in the last couple of decades, there's been a lot of work done here. And in today's talk, I'm going to touch upon both these types of models, both the contexts in which these types of models are used. So, before going forward, too, it's really important to emphasize how important salmon are as a key integral piece of the ecosystem, the marine ecosystem of the Salish Sea. So what happens to Pacific salmon between the time they leave the rivers where they were born and the time they return to spawn? A lot of that's not really well understood. I mean, we have a good general picture, but specifics of year to year or even decade to decade are not necessarily well understood. In addition to being very important parts of the ecosystem, they're also really important from a socioeconomic and cultural perspective. There are 5 species of Pacific salmon, and they're typically differentiated from trout based on whether they migrate to the ocean and whether they spawn just once or whether they spawn multiple times. So, coho and chinook here are of particular interest because they spend a relatively large amount of time in the Salish Sea as juveniles before migrating to the open ocean or other areas to rear and grow. And this is why in this research I'll talk about today that we've focused on coho and chinook. Over the past 40 years or so, coho and chinook have not survived as well as in the ocean. This is pretty perplexing, and nobody really knows what happened. The mystery was a motivation for this research and for the Salish Sea Marine Survival Project, which many of you were a part of and familiar with. It's a multi-jurisdictional project, a Canada-US multi-group, multi-institution collaboration. And these declines seem to be more similar among populations that are close by, like geographically close to each other or within the Salish Sea versus uh, populations of Chinook and coho outside the Salish Sea. The decline of coho and Chinook has also had a major impact on fisheries, not just recreational fishery, but here I'm just highlighting that fishery within the Strait of Georgia. So particularly, it has just been gutted, as you can see, by declines in productivity of coho and Chinook. So these issues, the marine survival issue, led to the Salish Sea Marine Survival Project, and scientists who worked on that project synthesized various hypotheses about what are the primary factors affecting coho and Chinook early in their marine life history. And one goal of this modeling work is to evaluate those hypotheses in one kind of integrated modeling framework. A second goal is to try as best as possible to recreate conditions that fish likely experienced in the Salish Sea in the past. And so this is the next part I'll talk about. This sort of leads into the model. There are 3 categories of model used here to form an end-to-end model framework. We use the NEMO oceanographic model for physics, and we use the Ecospace model with a custom submodeling framework for biogeochemistry and the ecosystem model, which includes fisheries. So I'm going to begin by talking about the NEMO model. This model has been developed and applied throughout the world, but recently researchers at the University of British Columbia's Earth and Oceans Sciences group, members of Professor Susan Allen's lab there, developed a NEMO model for the Salish Sea called Salish Seacast. And we were aware of this model, but it's very high resolution, and it did include biogeochemical components. And the high resolution of the model and the biogeochemistry made it a challenge to apply for a multi-decade hindcast. The model is run on supercomputing cluster, but even then there are computational limits. So I worked with one of Professor Allen's former postdoc researchers, Dr. Michael Dunphy, who's now a research scientist with DFO Canada. And we thought maybe we could trim the model down, like coarsen it a little bit or adapt it in certain ways, and just to make it work for our purposes of doing this multi-decadal hindcast. And Michael thought we could possibly speed it up by like a factor of 5 or 10. And so that's what we attempted to do. But I think before getting into the details of that, it helps to have a little bit more context as to why this is so critical for ecosystem modeling research. There's a long list of physical oceanographic processes and mechanisms that are of relevance for fishery science and ecosystem research. This figure is a great figure from a compilation of long-term environmental changes that are understood to be important. For example, like the atmospheric conditions play a huge role in biological productivity of the Salish Sea. Atmospheric conditions determine whether the temperature and the seasonal patterns of, or determine the temperature and seasonal patterns of river influx and the volume of river runoff and runoff just generally, and other aspects. So, for example, and the direction and strength of seasonal winds is known to be an important important factor determining the timing of the phytoplankton bloom. There are also these broad scale climate patterns and modes of variability that can determine how strong the upwelling is off of the coast. So this is a really important process because upwelling brings nutrient-rich, dense, cold seawater in at depth through a process called deep water renewal. And if upwelling patterns change, it can mean that these critical deep water renewal events also change. And then, of course, air temperatures are going to impact sea temperature, more so in the short term and at the surface. And water temperatures in the Strait of Georgia are warming twice as quickly as nearby open ocean. Around one estimate put it at 2.5 degrees Celsius per century. So it does sound like a lot of factors to be taken into account, but just to put it in context, um, and just not to 2.5 degrees might not sound like a lot actually, but it takes something like 3,000 or more than 3,000 times the energy to warm a volume of seawater than it does to warm an equivalent volume of air. So the max depth of the Salish Sea is over 400 meters, and the mean depth might be around 150. So it's quite striking that ocean temperatures over the entire water column are warming this rapidly. And there's indication that species are highly sensitive to what may seem like small ocean temperature changes. But not to mention the variability too. So it's also important, not just the secular trend in temperatures. So I'll touch upon that in a little bit. So luckily, to know a little better what's going on in terms of ocean temperatures over the whole water column, we have one station in Canadian waters at least called Nanu Station, and it's in the central part of the Strait of Georgia. And the Nanu's data are shown here as temperature anomalies in the plot on the left. And it's quite apparent just from this image that temperatures have increased over the past 40 years. So why do we need a physical ocean model if we know this to help recreate this? Well, the problem is that there are many gaps in our observations. So we just have this one Nanus location. And a reasonable question is, what does the picture look like in other parts of this area? Is it similar? The map on the right shows the Salish Sea, and the black star shows the Nanus station, and the various symbols denote where observations have been made over the last 40 years or 50 years. And there are a lot, but other than the lighthouse data that documents water properties near the surface, there are few, if any, time series from the Canadian waters other than this Nanus station. So we have a 2D vertical picture of water temps at this one location, and we want to run a spatial temporal ecosystem model. We could really use full coverage with respect to the water properties at these other locations throughout the domain. So this is where this physical model came in and the need for it. So we developed this NEMO model. We call it the hindcast of the Salish Sea, or hot sea. As I said, we simplified and modified a pre-existing model by coarsening it first. So it's 1.5 kilometers in horizontal resolution. And we had to remove the biogeochemical component of the Salish sea cast and smelt model. Those are the ones developed previously at UBC. And so we removed the plankton and the feedback loop between the biology and the chemical and physical components, just out of necessity. And the grid used is they're square cells, and in the vertical there are 40 depth levels. At the surface, they're spaced around a meter apart, but gradually stretched as the depth increases. The NEMO model uses a time splitting approach for efficiency. So there's effectively multiple temporal resolutions that range from 2 seconds in the vertical to 6 seconds in the horizontal or barotropic, like tides and waves, and the 2 minutes for the baroclinic, which are more kind of slower internal physics. The model gives us a temperature, salinity, tides, currents, other properties like measures of stratification. And it consumes a huge volume of data. It requires a huge volume of data. It wouldn't be possible to do this work without that. So this diagram just simply shows for like not all of the data, but how we really relied on the work of the previous work of researchers to make this happen. And so we really have to give credit for these various sources of data used as forcing or data used for evaluating the model. So before you apply a model to some use case, you should confront it with reality. We evaluated this model's performance using that huge library of data, and we focused this evaluation on Canadian waters just as a preliminary evaluation. So it was necessary to triage because we thought the volume of data and the just overarching research goal is to support the Strait of Georgia ecosystem model. But really, we were kind of skeptical this would really work at all. So before devoting more time to data collation from other sources, we just wanted to make sure this model worked. The evaluation is documented in our publication. It's been accepted. It's just in press right now, so I'll just point that out that you can go and read more there in the preprint. I think it should be out, the final one, within a matter of days or weeks. So we'll see. The highlights are that the model performed surprisingly well at recreating observed conditions at timescales of years, seasons, months, weeks, even days. Uh, of course, there are errors and biases in the model, but they're prominent at relatively high spatial resolutions. And so we're generally pleased with the result. For example, in the evaluation showed here, we see the mean temperature and salinity profiles over depth of the model versus observations. Our model is the red dashed line and the observations are the solid line. The black dashed line was an early version of the model. So I had no time to go deep into it, but we corrected for a source of bias, which improved the model substantially. And averaging over seasons and years, the mean error of the model for the bias is less than half a degree centigrade and around 1 unit of salinity, just uh, generally averaged. And then it performs best in open areas versus narrow topography. So the first row is the northern Strait of Georgia, the second row is the southern Strait of Georgia, and then we have the Juan de Fuca Strait in the bottom row. So we're happy with this, but whether the degree of error and bias is acceptable, it really just depends on the application of the model. And for our application, this was satisfactory. And in fact, surprising. So that was great. This plot shows the model bias over depth and month at nanostation specifically. And clearly the biases do have a spatial temporal component. So typically they're still less than half a degree at depths over 10 meters. In the top row, we have salinity. And in the far right, you see the model is too fresh in the summer. It's in the first couple of meters, between 1 and 2 standard units of salinity. In the second row, we see that at the surface, the model is too warm in the very top half meter in the summer and too cold in the winter. I say that's not a bad problem to have, but which is because our observations are biased towards surface measurements. So there are other sources of data for sea surface temperature and salinity, and we know far less about conditions 10 meters deep or greater. Anyway, there's certainly room for improvement, but we're really pleased with this. And the And critically, does the model recreate the trend I mentioned at that one location where we do have observations? So let's look at that now. Yes, it pretty well does. The model trend in red, it tracks fairly well the observed trend at nanostation. So statistically, the trend was the same. Um, we are currently limited by we missed the 1970s. So during which time there, you can see on this plot that the water temperatures were particularly cold. It was like a switch flipped or something around 1978, and temperatures in the strait increased quickly afterwards. But we're currently limited by the forcing data available, and we think this will change in the near future. But currently, we can't really cast backwards farther than 1980 due to limited data at the ocean boundary. So seasonal trends over depth, what about those at the same Nanus location? Here we see the seasonal trends. It's decadal trends over depth by season. The model's in red again. The observations are in black. The dashed line, the vertical line that's dashed, it indicates no trend. And the error bar denotes statistical significance. So the trends reported are weaker than previously reported because of the omission in the 1970s, but it's clear that there is a trend, and it becomes more clear that there are some areas for improvement that we couldn't see from the last plot. But especially in the summer between 10 and 100 meters. But overall, again, we're fairly surprised. The performance of the model here, it's fairly good. So it's not surprising that we see temperatures near the surface are warming relatively quickly, but the seasonal aspect is quite notable. So basically, the summer and the fall are experiencing the warming between the surface and around 30 meters. And what about elsewhere? Like this leads us to ask a question, what about elsewhere in the Strait of Georgia? So let's pan around a little bit. So it gets really interesting. I'm just showing the top 30 meters. These are maps of the central northern part of the Salish Sea. The gray hatching indicates trends that are not significant, and there's just quite a bit of information here. So for example, the model predicts that the waters in the winter might be warming in the 0 to 30 meter depth strata, but just not at Nanu Station. And waters in the spring don't show a discernible trend anywhere, though the stats we have indicate higher variability in recent years. And looking at the summer, it looks likely that the northern Strait of Georgia is warming fastest. And then the fall is really quite striking because significant fall temperature trends are quite widespread throughout the domain. And this could have important implications for juvenile salmon that migrate northward through Johnston Strait. And it could have implications for the variability, timing, and other aspects of the spring bloom or just fall bloom, and for zooplankton productivity. So what are the next steps? The hot sea model outputs, they do help fill a gap in patchy observations. These model outputs have already been used. Dr. Research scientist at DFO, Dr. Andrew Edwards, others and myself have already integrated model mean outputs, monthly mean outputs from hot sea into an integrated database called PASEA. This not only fills a gap in observations, but it fills a gap in model outputs. So we've been able to kind of slot it in there into the blank to fill in this gap for the Salish Sea. PASEA's data is used by stock assessment scientists and researchers investigating climate impacts on salmon. The data here are also being used in one project right now led by Dr. Cam Freshwater and Dr. Patrick Thompson, looking at Fraser River sockeye. And clearly the model ought to be extended further back in time. That's a next step. But an immediate next step is to apply the model in the end-to-end model framework that I mentioned. Let's see here. So returning to the end-to-end model framework, a typical way to do it is as shown here. It's called a three-stack setup, and we had to do a little improvisation. So the end-to-end model plan originally was this typical setup, but the biogeochemical model or the BGC model are coupled and linked in this way. They typically include zooplankton and phytoplankton, And the good things about the BGC in this kind of a setup is this feedback loop, nutrient cycling between physics and the biogeochemistry. It's also typically 3D. It's high resolution. It's often got the same resolutions as the physics, more or less. And ecosystem modelers like me don't have to do it. That's another thing. If this model exists, it can be very difficult to develop these models. They're very challenging. Um, a downside to them is they're computationally expensive. But so we didn't have one of these, so we focused, I guess, on what else we could do. We had to come up with an improvised plan. So we decided to improvise and try to use Ecospace in lieu of a traditional biogeochemical model. So this Ecospace model is part of a suite of ecosystem modeling tools along with EcoPath and EcoSim. And the model is the least developed. The Ecospace model is the least developed, but developed some time ago. And so I'll just call this a two-stack setup. But we did it in a modular way. So we have two versions of our food web model, this submodel 2 and submodel 3. And it worked. If it worked, the advantage of doing this is that the Ecospace BGC model will be fast. It'll be relatively cheap. It will be cheap computationally, but also with time and labor required to tune it and maintain it and develop it further. So another advantage that is substantial for people running these E2E frameworks with the Ecospace and EWE system is that it doesn't break the feedback loop between predators and prey. It does break it between the abiotic and the biotic at the base of the food web. So, but overall, this whole thing represents an experiment done in 2024. It's kind of a necessary improvisation to get to our overarching goal. So I call this a second piece of work, the problem with plankton. Having no plankton really is a problem, but how to do it is also a problem. It's very hard. And the geochemical cycling is really challenging. So as you can see from this image, the purpose of showing this image from this great compilation of effects of global and local change on the Salish Sea is to show you that there are many different processes, and most of them won't be captured by this model. But they're everything from pollutants to anoxic episodes to ocean acidification, carbon cycling. Suffice to say, it's a complicated picture, and no one model will be able to capture these processes. And there's also these biological processes. But one of particular and long-standing interest is what happens if the timing of the life history for a prey species changes, but it remains fixed, the timing of the life history for the predator species. So, for example, what if there's a mismatch between phytoplankton blooms in the spring and the timing of fish entering the strait or zooplankton hatching? So if phytoplankton blooms are timed differently, if they're more variable or early, it could mean that the dinner is not ready when these critters need to eat. So I decided this mismatch would be the focus of this experiment. Let's take a look at the spring bloom timing. What we know in the Strait of Georgia, Ecospace is a two-dimensional model. It's horizontally resolved but not vertical. We have relatively large gaps in observations here, but we do have a 1D model, and we have some satellite imagery data that's been processed. And from the model and those satellite images, it's been possible to develop an estimate of the year day of the bloom. So this 1D model of Allen and Wolf, it's set up for the southern Strait of Georgia. Again, it's kind of similar to Nanus, except it's a model where you have this one location and it predicts the spring bloom timing. But can we get the Ecospace 2D model to perform kind of comparably? The results of this previous modeling work, it indicates that there may not be a discernible trend in the spring bloom timing, but the variability in that bloom timing is becoming greater. It's becoming more unpredictable. So let us see what we have to but we have to capture kind of at the minimum in order to have a chance of getting this spring bloom timing to work in Ecospace. We have to know that the area is highly productive. It's turbulent with strong tidal and wind drifting mixing. Nutrients are essentially unlimited through the water column, except in the surface waters where they're depleted seasonally by plankton and by stratification. The Fraser River, it seasonally extends a silty plume across the domain. It limits light availability. Seasonal succession of phytoplankton is typical of a coastal mid-latitude area. And basically, you want to see the model create that pattern there you see in this cartoon, I guess. The light levels received by plankton and turbulent by plankton are primary factors, and the turbulent mixing is a primary factor that we need to worry about to get the spring bloom timing right. So I developed this diagnostic model in 2024. It's limited to the lower trophic levels, which it's got 21 functional groups from bacteria to juvenile fish. The model is carbon-based, so all groups are represented with units of carbon. The model was implemented using a custom framework to make Ecospace use a 3-day time step when it normally uses a 1-month time step. It's some satellite data are not resolved to resolution more than 7 days, so 3 days felt like the minimum we could possibly use. The maximum we could possibly use and get away with it. So I captured the plankton functional diversity and trophic interactions along these 2 dimensions of function and size, and estimated the maximum, minimum, and optimal prey size ratios. And you see that in the figure on the left. The figure on the right shows the food web. Phytoplankton and primary producers are in green at the bottom, and juvenile salmon and forage fish are at the top. It's just more details. I mean, the resulting food web, it explicitly represents the microbial loop and the so-called jelly web, which refers to this trophic network associated with jellyfish and gelatinous groups. Mixotrophy is also represented, and that's when a creature can use the energy from the sun via photosynthesis or through predation and respiration. So the takeaway here is that this is more detail and is usually folded into an Ecospace or EWE model. Another key aspect of this work is the pathways to juvenile fish. So here the key pathways were hypothesized to be shortest paths to juvenile salmon. They're shown in yellow. So these pathways are generally associated with spring bloom conditions. I'll have to move through this very quickly given the time remaining, but Ecosim and Ecospace do contain a way to simulate primary production and to do biogeochemistry. It's just not been explored very much, and it's a very simple model. The primary producer growth is model based on a saturating type relationship, which is kind of a textbook approach, and then the nutrients are depleted using a monotype equation. There's just one type of nutrient in the model. There's something called environmental responses, and you can do this in a couple of ways conceivably. You could do it in a kind of a crude way, and you can do it in a more precise way. And we use the thermal reaction norms and Q10 relationships to kind of represent the environmental response of phytoplankton to light and temperature. And so we have a method, and we'll have to move through this, but the purpose is it's just complicated. The system, this figure is a standard software diagram. It's a UML diagram. It sort of contextualizes what's being done to move between the 3D forcings to the 2D model. So this submodel here was custom developed to compute the light received by plankton over depths for each model grid cell. And then we're able to feed into Ecospace the required fields. So how does the model perform? This is really important. Of course, you got to confront the model and see if it works or it doesn't. And it seems to work pretty well at, these results are preliminary, but I can show here a comparison between the seasonal succession predicted between this Ecospace model and then a 3D model, the SMELT model for the northern Strait of Georgia. And then we compared them with the sampled biomass of diatoms from northern Strait of Georgia. We converted this to a normalized anomaly, and the in situ data were collected by the Hakai Institute and published. So it's great that we could use those data. And it's apparent that there are a few differences between the Ecospace model and the observations shown in pink, but the seasonal patterns are fairly similar. So this variability should be expected to be different because the model outputs are spatial temporal average, and the phytoplankton, the in situ data are collected from, on a given day and time, and phytoplankton are very patchy in their distributions. Let's take a look then now at the satellite data and the 1D model of Allen and Wolf, and we've extracted the ecospace predictions for this day of year of the spring bloom timing. On the y-axis is the day of the year, and the model, our model, the ecospace model shown in black, the 1D model in green, and the satellite predicted timing in blue. We do find good general agreement between the predicted bloom timing between 1980 and present. And there's certainly some deviations, but just draw your attention to the first 6 years. It's fairly close. And have kind of dug into this a little more. So I'm currently digging into this a little more to kind of understand when and why they do deviate. The horizontal bars, the gray bar and then the dashed line around it, indicate typical kind of average spring bloom timing. Anything below is early, anything above is late. Now, if we look at just the satellite data, an early run with Ecospace found in terms of the categorical timing of the bloom timing, we're getting it 13 out of 14 years for which there are data. So this is, very good. It's very encouraging. Now I'm just going to show you what is a bloom. What does diatom bloom look like in Ecospace? So I'm going to try to use this animation hack here to kind of walk us through. We see in the map the Ecospace model domain. It's kind of a crude map, but I'm working on a revision. There is an area immediately outside the Fraser River mouth, currently masked out there. It's kind of a blob because of the Fraser River plume. But January 2005, it starts out as typical. Conditions are not great for diatoms. The area is too turbulent, light levels are low. There is a hint of a bloom as conditions kind of momentarily look good in Boundary Bay, south of the Fraser River. But by late January, the weather shifts again and the winds turn. Then in February, there is a clear shift here. So in most turbulent areas of Harris Strait, we see mixing is shoaled and relatively high diatom biomass is seen. In February 19th, I'm already in March here, but if we go back to the 19th, we see the bloom is kind of in full force here. So we've reached a peak by roughly the end of February. And conditions are good, but nutrients are depleted and light and mixing levels reach values that are amenable to other primary producer functional groups other than diatoms. So as May progresses, let's see, nutrients are depleted, light, it's just not favoring diatoms anymore. So anyway, that is drawing my talk here to a close. I did look at spring bloom timing in the northern straits, similar to how we used the previous hot sea model. The trend was not significant, but what might be interesting here is it does raise the question: is the incidence or frequency of early blooms becoming more frequent, even if there isn't a secular trend? And the takeaways so far. So we can capture some aspects of plankton dynamics using a relatively simple model. Hot sea ecospace linking is successful. The model shows potential for driving the larger ecosystem model and evaluating these match-mismatch hypotheses. There's more to say, but it's about kind of the most ongoing work. So this winter, I'll be focusing on putting it back all together. So we kind of broken the system apart, put it together, see what we broke, fix it, and put it back together. The this bigger model is quite complex. It's got 69 ecological functional groups. There's still 3 being worked on. There's a technical report in prep describing this full E2E model, and we're using a modular approach to do it. And I see I've run out of time, so I'll just quickly go through this. Give me 10 seconds just to say that.
Yeah, no pressure, Greg. Take your time. This is like the important part. The what's next. So go for
Yeah.
it. Take your time.
Okay, I appreciate it. Thank you. Yeah. So yeah, I think the work so far has shed some light on changing environmental conditions that are relevant for Pacific salmon and for forage fish. I think that this NEMO Ecospace end-to-end model has promise. I think that um, there are trophic interactions, certain ones that I didn't speak about today. I have kind of got some, slides prepped, but we just had to go past it. But even if they're not focal on directly on juvenile salmon, they're really important to get right in these models if the predictions are going to be robust. And yeah, there's just some natural next steps. I think I think ultimately at the end of the winter and into the spring, we'll be getting at evaluation and synthesis of these hypothesized drivers of marine survival and juvenile salmon. So with that, I'll pause there or end it there, and like to say thanks very much to all the collaborators on this project and who've helped me out, and thanks again to the roundtable and organizers.
Thanks so much, Greg. That is a lot of information. I suspect there'll be some very keen modelers on the call today that will be putting their hands up. Right? Oh, look, there's already some people out there. I'm just gonna navigate I think it's Joel Baker. Go ahead.
Hardly a keen modeler. Sorry for the expectation. Thanks, Greg, for the presentation. I really appreciate the value of doing hindcast modeling. I think it really allows you to open up a lot of boxes about how these systems are working, and it gives you a lot of confidence in your forecasting models. If you can hindcast, then you should be able to forecast. I had a question about the early part of your presentation where you're showing the trends in temperature and salinity in the system in different seasons and how that grew in. I thought that was really interesting. I'm wondering if your modeling approach allows you to explore how much of that change is due to changes on the landscape versus changes in ocean condition. Thanks.
Yeah, thanks for the question. I can bring up that slide pretty quickly, I think. Yeah, it's for sure. Like this is, I guess, just like my my talk might have been a bit rushed. I'm trying to squeeze in a lot of info in a short period of time, and we're kind of trying to squeeze a lot of modeling in a short period of time. But I think with this model and with others similar to it developed in the area, you can do these model experiments. So it takes time and organization and funding and people to work on it, but you can isolate variables and run what if scenarios. We haven't done that to a large extent. We haven't done that, but when we were looking, this is the seasonal trend over depth at one location. If you look at this one, it's overall locations just in the surface 30 meters. But what we did to kind of get to this point is we had to identify a source of bias by doing these experiments. And when we did that, we isolated this pretty large effect of the temperature and the salinity of the water at the open ocean boundary. It has a big effect, even 200, or I don't know how many kilometers, but even all the way into the system. Like if you go up to the northern Strait of Georgia, we were seeing when we did bias correction to this water that is being kind of forced into the model at that boundary by the Juan Fuca Strait, that we could see this large effect on the temperature of deep water. So my suspicion is that that deep water renewal process, no surprise to any physical oceanographers familiar with the system, but I think based on that, that's just an example of a type of experiment that you can do. And another thing that needs to be done, I feel, is to experiment with Fraser River and other river water temperatures, which currently are not changing. They're basically a climatology. They're there is a water temperature. I should correct that. Yeah, I should go double check that, but I'm pretty sure that Fraser River water temperatures are increasing, but the flow rate is not changing. We just have we don't have the data together. So to evaluate the sensitivity of that would be valuable for sure.
Thanks. Hopefully that answered your question, Joel. There was a couple hands up that went down, which I suspect you might have answered their question. I'll jump in with one, and I am not a modeler. Just you're talking about the change in time over the impact of rainwater versus snow melt. So if you're hindcasting that, how does that affect your ability to forecast it as well? Is that something that you guys are able to quantify in this model and getting better at
Yeah,
tracking that?
yeah, good question. So there is for freshwater input, there, of course, we have rain, we have precipitation, which we get from, and snow, we get from these global climate models that are available to do projections with. What we need for freshwater, like river and stream input, is being worked on. There are research scientists at ECCC and at DFO working on downscaling the broad scale climate models so we can get like specific stream temperatures and use them for forecasting. And a workaround is we can create a climatology of flow, or we can actually have for the biggest river, we do have meters, like gauges that we're using data for Fraser River. And but so we kind of get an idea of how the river's flow has changed over time, like in terms of the timing and the whether it's snowmelt dominated or it's moving to more precipitation kind of dominated pattern. Uh, but yeah, so we basically need to have these forcing data available at kind of a higher resolution than are currently available, out of the box on these global models. And I know several people are working towards that for the interior BC rivers, at least, and I think for Vancouver Island too.
Yeah. I think Quetzino
Mm.
Sound, there's a couple.
It's
Um, I,
actually one of the reasons we were surprised it worked pretty well was because we didn't have better river forcing in terms of temperatures for all rivers.
nice. Okay, so we have a few minutes left. So I see Isaac's smiling face. Well, I can't quite see his smiling face, but I see part of his face. So I'll let Isaac jump in, and I think we have time for one more, depending on the answer with Jan, and then we'll wrap it up there. So Isaac, go ahead, jump on in.
Great. Hi, Greg. Thanks for a great talk. I just have a quick question about how you incorporated temperature into the response of your fish and your other vertebrates. I know there's been some interesting work trying to develop those thermal response curves in Ecospace, and I just wonder how you did it. You mentioned briefly something about a Q10 curve, but how did you put those pieces together? Thanks.
Yeah, thank you. Yeah, it's great to get I realize my talk was very technical, so I'm happy somebody wants to know that kind of stuff. Yeah. Yeah, this is kind of a diagram that shows some options for doing that. I could take where's my mouse here? Can you see my mouse? Okay. Yeah, so this this is kind of one example of a thermal reaction curve that's shows kind of an optimal range of temperature for a creature. And then there's a very, very simplified trapezoidal response that's commonly used in Ecospace and EWE and Ecosim. And the philosophy of the whole community that uses this software framework is generally to try to do it the simplest way possible, to actually do something crudely first, and then test the sensitivity of doing it more in a more detailed way. So I did use in this iteration of the model this simplified trapezoidal kind of response function, which it kind of combines the two concepts. It kind of combines the concept of the thermal reaction norm and then the Q10 relationship, because you just have this kind of optimal window. So that then I applied it there are many options to ways to apply it, of course, right? And I've only applied it to phytoplankton. I have not applied it to zooplankton at all. And there's a lot of great work, especially recently, to help inform the rest of the parameterization. So yeah, we're really kind of at an early stage here. Just wanted to see if just doing it with the phytoplankton would give us spring blooms that are fairly reasonable. We also don't have enough zooplankton data to really do statistically rigorous assessment of zooplankton anomalies or abundance. We have good data, but we just don't, and have enough. I think we need more. So yeah, that's just generally how I did it. I applied it as a foraging response. When it's applied to phytoplankton in this model framework, it it forces growth rather than forcing mortality or foraging. So that's how it was applied. The framework does kind of critical to this actually. Behind the scenes, the framework treats phytoplankton differently with these forcings than it does to consumer groups or predator groups.
Yeah, thank you. That's a nice, simple approach. Appreciate the explanation.
Thanks, you guys. Okay, so Jen, with our last question, and I'm just going to hold you with bated breath for a minute just to give a shout out to the upcoming, the next presentation. Muriel's posted it in the chat. It's the Tale of Three Auks, so just hopefully you can join in. It's going to be bird related, which is quite a right-hand turn from our typical fishy water stuff. But yeah, jump on in, Jen, with your question for Greig.
Thank you so much. Greig, that was a really great talk and excellent body of work. I was very intrigued by Joel's question, and I just wanted to make you aware of a different Nanus, not the place name. We have a monitoring buoy at the strait, the entrance to the strait. It's kind of off La Push, but it's getting the canyon water. And I'm putting a link in the chat. What John Mickett, who's the lead for this project, has been seeing in the depth profiles is right after the marine heat wave known as the blob happened, the very cold 8-degree water basically vanished. And we were thinking, because it's sort of each year the isotherms shifted, and we were thinking maybe this was a trend. But then in 2021, it came back with a vengeance. Anyway, all this to say, if you are looking for some data to test, kind of the hypotheses that you were exploring with Joel, be very happy to connect you with John, and we have all the data in ERDAP and all that good stuff. So great work, and really exciting to see the ability to test some of these hypotheses. So thank you so much.
Thanks, Jan. Yeah, I'll reach out.
Jen, that's a perfect wrap up to this of collaboration, sharing data, communicating back and forth. So